Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video I'm going to be discussing blood bank genetics. Alright, let's get started. If you are in a medical laboratory technology program, you likely have already learned about genetics. It's a very important concept crucial to understanding immunohematology, which is also called blood banking. Genetics is the scientific study of heredity or inheritance and the variation of organisms. It is how traits or genes are passed from parents to offspring. Chromosomes are thread-like structures within the nucleus of cells that carry genetic information. They're made up of DNA and proteins and come in pairs. Humans have 23 pairs of chromosomes, or 46 in total. The term diploid refers to two complete sets of chromosomes found in cells. 22 pairs of these chromosomes are called autosomes, and one pair are called sex chromosomes. So autosomes are any chromosomes that are not the sex chromosome pair, and the sex chromosomes are either X or Y. Before we get started, it's important to review some definitions that relate to genetics. So locus refers to a position occupied by a particular gene on a chromosome. An allele is any alternate form of a gene that can occupy a given chromosomal locus. Homozygous refers to when a pair of identical alleles at any given locus, and a heterozygous refers to when there are different alleles at a given locus. Genotype is the genetic makeup of an individual, whereas phenotype refers to the outward expression of genes. So I will give examples of homozygous versus heterozygous and genotype versus phenotype later on in this presentation. The term codominant is when both alleles contribute to the phenotype. An amorph is considered a silent gene that doesn't produce a detectable trait. So now we can't talk about genetics without talking about the father of genetics, Gregor Mendel. Mendel was an Austrian scientist who conducted research in a monastery's botanical gardens in the late 1800s. Discussion of his work is discussed in almost every single biology course you can take, so I'm not going to be going into detail here, but essentially he was responsible for the proposal that genes occur in pairs and that one of each gene is passed from parent to offspring. Punnett squares are a visual tool used in genetics to predict the possible genotypes of offspring from a cross between two parents. The use of Punnett squares helps to illustrate the likelihood of different combinations of alleles being inherited and expressed in offspring. All right, so let's look at some Punnett square examples. So again, Gregor Mendel used pea plants, and I remember in my undergrad we would talk about pea plants and the wrinkled and the smooth pea pods and what color they were and all this. So honestly, I found that really boring. I personally am a dog person, and I actually find that the color genetics of purebred pugs is actually a very easy example of how to use a Punnett square. Now, if you all are upset that I didn't use pea pods and use cute little dogs, I guess that's on you, but we're going to use cute little dogs to explain this. All right, so in purebred pugs, there are only two color types. So you can either have a black purebred pug or you can have a fawn purebred pug. Right? So the color black is the phenotype and the color fawn is the phenotype. Okay. Now genotypes are different, but phenotypes is what you see, what it expresses, right? So black is considered dominant, and the allele for the black color is big B. Fawn is considered recessive, so little b, okay? So for this dog on the left, it's a black pug. It's a cute little guy too. There are two possible genotypes for every single pug that is black. They can be big B, which is homozygous for B, homozygous meaning that they have the same identical alleles, or heterozygous, big B, little b, okay? So in this particular instance, big B It's of course going to be black. It's homozygous. This heterozygous big B, little b, it would be considered a carrier for fawn because that little b is recessive, right? But it's still going to have phenotypically express itself as black because that big B is dominant for the black color. Now for the little fawn here on the right hand side, this kind of like a brownish tannish color with a black mass, that's considered fawn. There's only one possible genotype for this, and that is homozygous for little b. So that means it has two little b alleles. Okay, so this is going to be our first Punnett square example. So over in the bottom right hand corner is a Punnett square, and I'm going to go step by step and show you how to fill these out. So again, we're going to be talking about pugs first, and then I'm going to relate it to blood banking. So recall again that the capital B or big B is a dominant gene, and it codes for the phenotype black in color in pugs. And then little b is a recessive gene, and it encodes for the fawn color in pugs. So in this example, we're going to take two black pugs, okay? So the black is the phenotype, remember, and the genotype, we don't actually know what the genotype is just by looking at the pug, okay? We would have to know its genes. But in this particular example, we're going to have homozygous black. So that means, homozygous means that it has the same gene on each allele. Okay, so each gene has two alleles here, big B and big B, big B and big B. All right, so both of these pugs are homozygous for big B. Okay, so what we're going to do on this Punnett square is we're going to take this first pug, this first big B, and we're going to put it in the top left-hand corner of the Punnett square. Then we're going to take the other allele, which happens to be big B as well, and we're going to put it on the bottom left-hand corner of the Punnett square. So that is one example of a parent. So they're either going to be able to donate one of those alleles to the offspring. It just so happens that these are both big B. Now let's look at the other parent here. So again, that first allele is going to go in that top left-hand corner of this Punnett square. And then the other allele option, which happens to be a big B as well, because remember these are homozygous, goes into the top right-hand corner of the Punnett square. Okay, so again, this is used to determine or give an idea of the possible offspring that this genetic pairing can produce. So let's do this top left one. Big B, big B. Okay. Bottom left hand one, big B, big B. Now remember, this is coming from this combination and this combination. So this one is here and this one is here. Okay, let's do it on this one. This possible gene combination there, and it's coming from here. Okay. All right, so let me get rid of that little pointer here. Okay, so what is the next one? Let's see if I can get off this highlighter. Okay, there we go. Okay, so this top right-hand corner, again, is going to be big B, because we're using this one and this one, okay? So the next one we'll do in yellow is going to be this B and this B. And it, again, big B, big B. Okay, so the genotype of all of the offspring, what are the chances of those genotypes? They're all big B, correct? So 100% of the pairing or the offspring of the pairing of big B, and big B are all going to be big B. Okay, so 100% are going to be homozygous for big B. What about the phenotypes? Remember, keep in mind that big B is black, codes for black, and little b codes for fawn. So if we have two big B alleles, what is that going to give us? That's going to be 100% black. So there's no chance of any of the puppies resulting from two homozygous big B black pugs. None of them are going to be fawn. Okay, let's look at the next example. Okay, so let's look at a different Punnett square example. So in the last slide, we bred two black pugs together. And both of those black pugs were big B, right? So were those homozygous for big B, or are they heterozygous for big B? Right, so the answer to that is they were homozygous for big B because they had two alleles that were both big B. So the difference in this example is we're going to use one homozygous big B parent and combine it, or produce the offspring of the mating of a heterozygous big B pug. Okay, so both of these pug parents are black in color, so that's their phenotype, and how they different from each other are their genotypes, right? So one of them is big B, and the other one is big B, little b. So this pug here on the left, okay, appears black, and the genotype is big B, little b. So why is that pug black in color and not fawn in color? Because it has the genes for both of them, right? So remember, big B is black and little b is fawn. So why is it black and not fawn? So the reason for that is that big B is dominant. So black is dominant. So that means if there's a big B present in the genotype of a pug on this specific locus, it's going to be black in color. Right? So, okay, so let's combine, using our Punnett square here, these two parents to see what the possible genotypes of their offspring are. So this pug, this first pug here on the left-hand side, one pug parent, is going to donate either a big B or a little b. And so we're going to put that on the Punnett square. Now this pug on the top, this pug parent, only has big B to donate, right? So we're going to put big B and big B on the Punnett square. So the first location, the first possible offspring is going to be in this box here, okay? Let's get rid of it so that way you just know where I'm at. Okay, so what are the possibilities in this little area? So big B and big B there, right? So that's going to give us big B, big B. All right, so let's do the one right below that. So we have a little b and a big B, right? So what is that going to give us? Big B, little b. Good. All right, so then let's do the top right-hand one. So we have a big B and another big B. So that's going to give us big B, big B. And then the bottom right-hand corner, we're going to have little b and big B. So that's going to give us big B, little b. All right, good. So this is how Punnett squares work. All right, we're going to do a couple more examples for pugs here, but hopefully you're getting the hang of it here. So genotypically, what are the percentage likelihoods of big B, big B? So how many possible puppies or possible offspring are going to be, what's the percentage chance that each puppy is going to be big B, right? So if you look, we have 1, 2 out of how many? 4 possibles, right? So that's going to be 50%. And then what's the genotype possibility of big B, little b? So what are the chances that each puppy or each offspring is going to be big B, little b? 1, 2 out of possible 4. So it's 50%. So the genotype of the offspring, 50% of them are likely going to be big B, which is homozygous for big B. And the other 50% is going to be big B, little b, right? So that is heterozygous, okay, for that particular gene. Now, phenotypically, what are all of those offspring going to be? What color are those puppies going to be? 100% of them are going to be black. Why is that? Because remember, black, capital B, big B, is dominant. So that means if any time there's a big B present, that particular dog is going to be black. Okay. Okay, let's do another Punnett square example. So in this case, we're going to be breeding two black pug parents together. And the difference is not phenotypically, right? So phenotypically, they're both black. But the difference between this one and the last two that we did is genotype, right? So their genotype in this particular situation, both parents are heterozygous, meaning they have one big B allele and one little b allele. Okay, so each of those are the same genotypically and phenotypically. So I am not going to do the arrows this time, so hopefully you're following along and understand it by this point in time. However, we are going to do this Punnett square together. So this first parent, this one on the left, has either a big B allele to give or a little b allele to give. And then this top parent, that is again heterozygous, has either a big B to give or a little b to give. All right, so let's do this Punnett square together now. So this first box up at the top, upper left-hand corner, we have a big B and a big B. So what are we going to get? Yep, a big B slash big B. Now the one right below it, we have a little b and a big B. So it's going to be big B, little b. Now the upper right-hand corner, one allele from the first parent is going to be big B, and the other allele that's inherited from the second parent is little b. So again, we're going to have big B, little b. Okay, now this bottom right hand corner, here's where we have a difference. Okay, so in this particular case, we have a little b from parent on the left and another little b from the parent up top. What are we going to have here? Little b, little b. Okay, so genotypically, what are our likelihoods of each puppy having each genotype? So how many potential puppies, or I guess that's kind of not the right way to word it. So each puppy has this amount of chance to be this genotype, okay? So it's not the entire litter. Each offspring has this chance. So in this Punnett square here, these 4 boxes in the Punnett square, how many of them are big B, big B? Just one, right? 1 out of 4 chance. So that means each offspring resulting from this breeding has a 25% chance of inheriting the genotype big B, big B. Okay. So what about big B, little b? There's 1, 2 here. So 2 out of 4, which is 50%. So each offspring out of this genetic pairing has the genotypic percentage probability of 50%, because 2 out of 4 possibilities of being heterozygous, meaning big B, little b. Okay, so let me erase here. Hopefully it doesn't confuse you. Okay, now what about little b, little b? Right, there's just 1 out of 4, so that's 25%. So 25% chance of being little b out of this pairing, and that's of course genotypically. Okay. So the genotype of the offspring, so 25% chance of being big B, so homozygous big B. Each offspring has a 50% chance of being big B, meaning heterozygous, and then a 25% chance of being little b, which is homozygous for little b. Okay, so phenotypically, now think real hard here for a second. Phenotypically, what is this going to be? This pairing. What is the percentage of this pairing? Right? So are they all going to be black? No, they're not. Remember the last 2 slides, we had 100% chance of every puppy resulting from them is going to be black, right? That's going to be different here. So how do we figure this out? So again, remember, big B is dominant, and that means if a big B is present, the puppy is going to be black. Now, little b is recessive, so fawn is recessive, and the only way that fawn can be expressed phenotypically is if this big B is not present, okay? So this one here, big B, it's going to phenotypically express as black. Big B, little b, what is that going to be? It's going to phenotypically express as black because that big B is present. Now this 25% one, little b, is there a big B present? No, there is not. So that means every puppy in this particular pairing, or every offspring in this particular pairing, has a 25% chance of being fawn. So 75% chance of being black and 25% chance of being fawn. Okay, so this is going to be my last example of using these cute little pugs, and hopefully by this slide you kind of know how to use a Punnett square and understand kind of what we're talking about. Okay, so in this particular instance, in this breeding, we are going to breed a homozygous black pug. So homozygous refers to the genotype, so that means it's 2 alleles of that big B, okay? And the phenotype of that pug is, of course, black. And then we are going to breed it to a phenotypically fawn pug. So the phenotype is fawn, and the genotype is homozygous for little b. Okay, so it's homozygous recessive, meaning it has 2 little b alleles. All right, so again, I'm not going to use the arrows here. We should know by now what in the world's going on. So on the left-hand side here, this parent only has a big B allele to give, okay? And I clicked a little bit here, a little too early, but in this first box that I just populated, it's going to be a big B allele mixed with a little b allele, and that top parent only has 2 little b's to give. So it can only give a little b or a little b, okay? So all of these are going to be big B, little b, okay? So genotypically, what are the percentages of the offspring? So 100% are going to be big B, little b. Now, is that homozygous or heterozygous? So the answer is heterozygous, okay? It's got 2 different alleles, okay? Now, phenotypically, what are they all going to be? We have a fawn parent now, bred to a black parent. Are they going to, some of them going to be fawn, some of them going to be black? No, right? Because that the kicker here is that the black pug parent is homozygous for black. So because of that, all of the offspring are going to be big B, little b, which means 100% of them are going to be black. Now, if you bred a fawn pug to another fawn pug, 100% are going to be little b, right? Also, if you bred a heterozygous black pug to a fawn pug, some of those offspring would be in fact fawn. But in this particular example, this black pug parent is homozygous for black. So it can only give that big B allele to any of its offspring. Now that we've discussed Punnett squares, let's talk a bit about inheritance patterns and their examples. Gregor Mendel determined 3 laws of inheritance. The first one is the law of dominance. This means that in a cross of parents who are pure for a contrasting trait, only one form of the trait will appear in the next generation. The hybrid offspring will exhibit only the dominant trait in the phenotype. So in simple terms, what exactly does this mean? So it means that recessive traits are always dominated or masked by the dominant trait. So in our pug example, right, the black color is dominant and the fawn color is recessive. If the dominant gene, so in this particular case, big B in the pug example is present, the recessive gene, the little b in our example, will be hidden or masked. Mendel's second law of inheritance is the law of independent segregation. This law is based on 4 concepts. A gene exists in more than one form of an allele. When gametes or sex cells are produced, the pairs separate, leaving each gamete with a single allele. Every organism inherits 2 alleles for each trait, one from each parent. The third law of inheritance is the law of independent assortment. This law states that genes are inherited independently from each other. In other words, the allele a gamete gets from one gene does not influence the allele received for another gene. The Hardy-Weinberg principle is an important concept in population genetics. It helps scientists understand how genetic variation is maintained or also changed within a population over a period of time. This principle only applies if the population is large. There is random mating, no mutations present, no natural selection, and no gene flow. The principle is expressed by an equation. P squared plus 2pq plus q squared equals 1. P is the gene frequency of the dominant allele, whereas Q is the gene frequency of the recessive allele, and 2pq is the frequency of heterozygotes. Let's talk briefly about inheritance patterns. Autosomal dominant, autosomal recessive, sex-linked or X-linked dominant, and sex-linked or X-linked recessive. So think back to the beginning of this presentation when I said autosome. Remember, these are chromosomes that are not sex chromosomes. In autosomal dominant inheritance, the trait affects both sexes. If a parent is affected and the other parent is not affected, 50% of their offspring will be affected. In this inheritance pattern, it cannot be transmitted by an unaffected parent, and the trait can also appear in every generation. This example on the right in this drawing, let's say that the affected means a person with a genetic disorder. The father is affected, and that affected gene is on an autosome. So again, not a sex chromosome. The mother is not affected. So 50% of their offspring will be affected with that disorder, and 50% will not. And notice here that it affects both sexes. So every child from this specific pairing would have a 50% chance of being affected regardless of their sex. In autosomal recessive inheritance, again, the trait can affect both sexes because it's on an autosome, it's not on a sex chromosome. The trait may appear in children, but not in the parents. So this occurs if both parents are carriers for the particular disorder or trait. So let's look at this example. Both of these parents carry the affected gene. 25% of the offspring will be affected, 50% will be carriers, and 25% will be unaffected. Now those are autosomal, meaning they are on chromosomes that are not sex chromosomes. The next two inheritance patterns occur on the sex chromosomes. So in sex-linked or X-linked dominant inheritance, an affected father transmits the trait to all daughters, but no sons are affected. And this is because a genetic male only has one X chromosome. Remember, he's X and Y. So he can only give a female offspring his one X chromosome. And if he has an affected chromosome, a sex-linked chromosome, it's only going to be on that X. So it's always going to go to the daughters. So on this picture example, we have an affected father and an unaffected mother. So you see there are no affected sons. Why? Again, because the father gave the sons a Y chromosome and the issue is on his X chromosome. In sex-linked recessive inheritance, this affects males. Affected fathers transmit the trait to all of their daughters, and those daughters transmit the trait to half of their sons. So daughters are carriers of the trait genetically, but do not demonstrate this trait. The trait is never transmitted from father to son. Again, why is this? So remember, it's on the X chromosome. So a father can only give his sons a Y chromosome, so they will be unaffected. So an example of this inheritance is a disorder called hemophilia A, which is a genetic bleeding disorder. So the picture on the right-hand side of the screen shows a pairing of an unaffected father with a carrier mother, and then another pairing with an affected father to an unaffected mother. Now, you don't want to really deep dive and get lost in the weeds with this, okay? So this is a challenging course, so blood bank is very challenging and has a lot of concepts to understand. So don't go too crazy memorizing all of this. Just understand the basic concepts of these. So how does all of this apply to blood bank? So understanding blood group genetics is important in transfusion medicine. It's actually critically important. So most antigens, which are present on the surface of the red cell within the blood group systems, follow a straightforward inheritance pattern. They're usually codominant, meaning both alleles contribute to the phenotype. I discussed the ABO blood group system in another video, so please, after this video, go check that one out. But the entire point of needing to understand genetics for the purpose of blood banking is for this right here. I'm sure all of you have heard people talking about their blood types, so usually what they're referring to is their ABO blood type. So there are 4 ABO blood groups: Group A, Group B, Group AB, and Group O. Now remember back to my pug example. The phenotype is if the pug appears black or fawn. So when someone says they're, for example, Group A or Group B blood, that is their phenotype. And just like with the pugs, there can be different genotypes for each phenotype. So for Group A blood, so the phenotype is A, they can either be A/A or A/O. A person with an A/A genotype inherited one A allele from one parent and another A allele from the other parent. A person with A/O genotype inherited one A allele from one parent and an amorph from another parent. O is considered a silent amorph. It just means that there is an absence of an A or B allele. For the group B phenotype, the possible genotypes are B/B or B/O. Now for group AB or group A, B, there's only one genotype, and that is A/B, meaning they inherited one A allele from a parent and the other parent gave them a B allele. And then there is group O, meaning they have no A or B alleles, and their genotype is O/O. So again, I go into more detail on this when I talk about the ABO blood groups in a different video. However, you need to know this, memorize this, burn this in your brain, okay? This is very, very important, all right? Okay, so we did pug Punnett square, so now we can do something relevant, blood bank Punnett squares. I chose the pugs because it's a little bit easier to explain than blood bank. All right, so we are going to have a mixing of a AA parent and an AO parent. So this AA parent, that is the genotype, right? So phenotypically, what are they? They are a group A person. This AO parent, that's their genotype. Their phenotype is what? Group A as well. So one parent is group A, other parent is group A, right? So let's do the Punnett square here. So here on the left-hand side, that first A is going to go there on the left side of the Punnett square. And then that second A is going there. Then up top, that AO parent can either give an A or nothing. So 0, the O, which is the absence of A or B antigen, right? All right, so this first one, what's it going to be? So we're talking about this one and this one here. All right, it's going to be A/A. Then the bottom right under that, what is it going to be? A/A. All right, now let's do this far right one up at the top. Okay, so we're looking at, let's do yellow here, this A and this O. What's it going to be? A/O. Then we're going to do this bottom, far right bottom tile. It's going to be A and O, right? There we go. Perfect. Okay, so what are the possible genotypes that result from an AA parent having offspring with an AO parent? So we have 1 and 2 A/A's out of 4, so that's 50% A/A. Let me get rid of that. And then we have 1 and 2 A/O, so that's 50% A/O because there's a total of 4 possibilities. Okay, let me delete here. Okay, so we're going to have 50% A/A and 50% A/O. All right, so those are the genotypes, right? What are the phenotypes? So think back to our pug scenario where the phenotypes were black or fawn. Okay, what are the phenotypes here? Are they group A? Are they group B? Are they group O? Are they group AB? Right? So all of these resulting offspring are going to be phenotypically expressed as group A. So 100% of them are group A. Okay, so let's do another one here. In this particular problem, we are going to have the genotype A/A parent. So phenotypically, they're going to be group A, and a B/O parent. That's genotypically, of course. Phenotypically, they're going to be what? A B/O would be what? It would be a group B person. So again, over on the left-hand side, we're going to put our A/A parent, and up at the top, we're going to have our B/O parent. So we're going to distribute these on the Punnett squares. Should know how to do these by now. And that first upper left-hand corner one is going to be A/B, right? Because we're taking this A and this B. All right, that bottom left-hand corner is going to be the same thing, right? A/B. Let's do that top right-hand corner. It's going to be a combination of this A and this O, correct? So what's that going to give us? A/O. And then the bottom right-hand corner, again, A/O. So genotypically, we are going to have 2 out of 4 A/Bs, so that's 50% A/B, and 2 out of 4 A/Os. So 50% AB, A/B, 50% A/O. Okay, now, phenotypically, how are these going to express? So the A/B, they have both genes, right? A and B. So 50% are going to be A, B, group A, B, or group AB. And the other 50% are A/O. So how does that present? Is there a group AO? No, there isn't. There's group A, group B, group A, B, and group O, right? So it's going to be 50% group A and 50% group A, B. Okay, let's do one more. So this one is going to be a group B parent that happens to have the genotype B/O, and a group O parent, which of course there's only one genotype for group O's, and that's going to be O/O. Right? So again, on the left-hand side, we're going to have the B/O parent, and we're going to distribute each of the alleles that it could give, and then up at the top, O/O, it's an absence. Remember, these are silent amorphs. This is the absence of an A or a B. So that top left-hand corner is going to be B/O. The bottom left-hand corner is going to be what? O/O, right? Because again, we're doing here and here. Okay, let's do that top right corner there, B/O. And then what's that bottom right-hand corner going to be? O/O. All right, good. So you should be able to do these by yourself by this point. So what's the genotypes of these potential offspring? So again, 50% is going to be B/O. So there's 1 and 2, right? And there's a total of 4. So 2 out of 4 is 50%. So we're going to have, and then O/O, again, 2 out of 4, so it's going to be 50% for that. All right, so we have 50% B/O and 50% O/O, right? So what are the phenotypes going to be? So those are the genotypes. So the phenotype, so a B/O is going to express as a group B patient, right? So we have 50% group B, and then an O/O is going to present phenotypically as a group O. So we have 50% group B, 50% group O. All right, so that's the last Punnett square and blood bank I'm going to do. However, I cannot express this to you enough. You absolutely, by the end of this lecture, need to know and understand how these alleles are being inherited. You should be able to do these Punnett squares like the back of your hand. It is critically important. If you're in my class and you do not know how to do these by now, please, please, please reach out to me. You don't want to go any further in this class without understanding how this works, okay? If you're not in my class and you are a fan of my YouTube, thank you, by the way. I really appreciate all the support. But if you are not in my class, please leave a comment. Ask me. If you're in blood bank and you don't understand this, this is critical at this point in time that you need to know how to do this. in order to progress. So either contact me as a professor or contact me via YouTube. You don't want to go any farther if you do not understand these concepts, okay? It's okay if you don't understand them. I'm happy to explain it in a different modality for you, but you absolutely have to, at the end of this lecture, need to know how to do these, okay? It's critical to your success in blood bank. It gets significantly more challenging, and this is the bread and butter, start of it. Alrighty, so if you like this video, go ahead and give it a like and make sure to subscribe to my channel for more educational laboratory content. Until next time. Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video I'm going to be discussing blood bank genetics. Alright, let's get started.